everybody, my name is Yasha Chapman, and I've had the opportunity to work in the Hofstetter lab. Today my presentation will be on methylprednisolone increasing tissue sparing after cervical spinal cord injury in rodents. So let's start off with some background terminology. A spinal cord injury consists of a two-step process. The first step being the primary mechanical injury, which consists of cells and tissue being destroyed by the initial impact. This is followed by the second step, the secondary injury. This, which can look like, in which tissue, this can look like, oh my gosh, I'm tripping. I'll restart just a second. A traumatic spinal cord injury consists of a two-step process. The first step being the primary mechanical injury in which tissues and cells are destroyed as a result of the initial impact. This can look like fractures, lacerations, contusions, and the second step is called the secondary injury. This happens over a few hours to weeks after the initial impact. And the secondary injury is characterized by inflammation, edema, and further expansion of the primary injury site. The secondary injury is something we would like to minimize in order to prevent as much neuronal deaths as possible. The main drug I will be talking about today is methylprednisolone. Methylprednisolone is a steroid which naturally occurs in the body. Oh my gosh, I'm sorry. Functions to the main drug I will be talking about today is methylprednisolone. Methylprednisolone is a man-made version of a steroid that naturally occurs in the body. Methylprednisolone specifically functions to decrease aquaporin 4 expression in astrocytes and inhibits apoptosis. Currently, the drug is listed as a reasonable option to patients that present within 8 hours of acute spinal cord injury. However, the drug is not standard of care and seen as controversial. A laminectomy is a type of surgery in which a surgeon removes part or all of the vertebral bone, the lamina. According to the National Spinal Cord Injury Statistical Center, there are approximately 18,000 new cases of spinal cord injury each year. 50% of these spinal cord injuries are in the cervical region. More than 90% are due to a traumatic nature. This could be due to incidences like traffic accidents, falls, sports, or violence. Which is why we focus on traumatic spinal cord injuries in the cervical region in our project. Unfortunately, there is currently no treatment strategy to restore spinal cord neurons and circuits after traumatic spinal cord injury, which is why there is a tremendous need to develop therapeutic strategies for these patients. Materials and methods. We use 20 8 to 12-week-old female long Evans rats, and the surgeon then introduced an incision over the C3 to C7 vertebrae. They then received a laminectomy to expose spinal cord level C5, and we then gave them we then set the Infinite Horizons impactor to deliver an injury at 200 kilodaltons. Treatment. So we had 2 sham animals which received a laminectomy only, so no spinal cord injury. We had 8 spinal cord injury only animals, 5 spinal cord injury plus methylprednisolone animals, and the last group I won't be talking about, but I put it in to show you guys that we used 20 animals in total. The rats received 4 intravenous boluses of methylprednisolone after injury. The first one immediately after the injury, the second one 2 hours, the third one 4 hours, and the fourth one 6 hours after the injury. The spinal cord injury only animals were given saline instead of methylprednisolone, and the rats received pain relievers, antibiotics, and softened food and hydration gel at an accessible height. Results and behavioral analysis. The behavioral testing was done 4, 7, 10, and 14 days post-injury. And if we look at the graph, you see that the laminectomy only animals had perfect scores because their spinal cord was not injured. These scores were out of 20, and we looked at the joint and ligament mobility of the animals and assessed them based on their performance. The spinal cord injury only animals and spinal cord plus methylprednisolone animals started off around 6 points and then improved to what you see now. However, if you look at the image, you see that there are no significant differences between the spinal cord injury only animals and spinal cord injury plus methylprednisolone. This could be possibly due to the fact that the experiment was 14 days long, and maybe if we extended the experiment, we would see greater differences. This is a cross-sectional slide. On the left we have uninjured spinal cord tissue, and on the right we have injured spinal cord tissue. Both slides were stained with GFAP, which is a protein found in astrocytes, and astrocytes are cells that can be found in the spinal cord and brain, where they perform multiple biochemical functions and also keep homeostasis. And in red, we see the macrophages and monocytes stained with CD68. The macrophages and monocytes perform immune system, are white blood cells that remove dead tissue cells and also microorganisms. And that is that. And then the next image I will be talking about is tissue sparing. If you look at around 0.5, you can see that the spinal cord injury only animals had less tissue sparing than the spinal cord injury plus methylprednisolone animals. The laminectomy animals did not receive a spinal cord injury, so therefore they had 100% tissue sparing. The next slide is quite similar to the last slide. However, here we focus on lesion area. And as you can see, the p-values show that the spinal cord injury animals compared to the spinal cord plus methylprednisolone animals, the difference between the values, the results show a high significance of 4 stars. And I want to thank Dr. Ellenbogen, Mrs. Ellenbogen, and Mr. Pritchett, and everybody who made this possible. And thank you for the memory.